All right, the purpose of this, let me turn off my, actually, no, never mind. It's not going to record the fact that I'm on video. Okay, the purpose of this video is to give you guys an opportunity to see the types of questions that are going to be on Friday's quiz. Remember, these questions are harder because you don't see the answer. There are a total of 1, 2, 3, 6, 10, 13, 16, 23, 6, 30, 34, and 7. There are 41 total questions represented in this. I encourage those of you that are here to keep track of how many questions you can get right, or you get right, especially how many questions you can get right without actually looking them up. All right, here we go. I'm going to need to step away in just a moment. I've got to go turn my my pool filters on, and I'm not using the timer right now, so I'm going to have to go turn that off in just a second. Okay, so I'm going to jump into a question that's going to take a little bit more time. All right, here we go. All right, let's see. What happens to the surface area or volume or surface area volume ratio when a cell size increases? True or false? Cell division increases the mass of the original cell. Before a normal cell becomes too large to carry out normal activities, it will usually divide to form two blank blanks. All right, I'm going to run outside real quick. I will be right back. Come on, pup. All right, you guys, sorry about that. Okay, here we go. Answers forthcoming. Okay, let's get this out of the way. All right, so the surface to volume ratio decreases as the surface area, as the cell size increases. My bad. And false, cell division does not increase the mass of the original cell. And daughter cells are what are produced by, of course, mitosis and meiosis. All right, here we go. Well, we'll jump into these. True or false? As a cell becomes larger, its volume increases faster than its surface area. Very related to the question you just saw. Okay, so I'm going to give you guys 30 seconds on these one question. Okay. About 10 seconds. All right, here we go. That's true. As a cell becomes larger, its volume increases faster than the surface area. Okay, mitosis question. DNA is replicated during which phase of interphase? Remember, there's the G1, the S, and the G2 phase. 30 seconds. All right, here we go. That is the S phase. Remember, in the Amoeba Sisters video, they said that it's called the S phase because of synthesis. Synthesis means to make things, and DNA is made during DNA replication during the S phase. However you want to remember it is totally up to you. Here we go. Contact with blank can stop normal cells from growing. All right, here we go. Contact with other cells. Remember, one of the warm-up questions we did suggested that when cells bump into each other, they know it's time to stop growing because there's no more room for the cells. All right, 2-point question. So I'm going to give you guys a minute for this. Why is crossing over important? What benefit does it give a species? Okay, that's an hour. We don't want an hour. We want a minute. My bad. Wish I could have you guys listening to what I'm listening. I got a rockin' 70s playlist. I love the 70s. But if I have the music playing for you, it's going to show up in the video, and then the video can't be on YouTube. So, sorry. So if you see me head bombing and moving around, that's what I'm doing. Okay, about 30 seconds. All right, here we go. Okay, it leads to genetic variation, which basically means differences within the population. Remember, population, same species, same time, same place. Human population, crow population, a rabbit population, whatever the case may be. Differences in their appearances, differences on their internal structures and processes too. A combination of new alleles, chromatids gets exchanged. The reason why genetic variation is important is because it leads to new characteristics, and those new characteristics might enable the species to survive if they have a changing environment. And of course, our environment is changing. Okay, as the cell becomes bigger, more demands are placed on what part of the cell. The larger a cell becomes, the blank, more or less, efficiently it is able to function. All right, here we go. All right, so it places more stress on the DNA or on the nucleus. Of course, that's where the DNA is found. And the larger a cell becomes, the less efficient it is able to function. Okay, remember in the first video we saw back to the Bozeman science, where you had you put the pieces of bark in the plastic cylinders. The cell has this happy place, okay? The right amount of volume, the right amount of surface area to be most efficient. Here we go. Blank division of the blank is division of the nucleus, and blank is division of the cytoplasm. These are the two stages of cell division. All right, here we go. Mitosis, division of the nucleus. Cytokinesis is the division of the cytoplasm. And remember, this equates to both mitosis and meiosis. You still have to divide the cytoplasm in meiosis. You just have to do it twice. Okay, so it could be mitosis or meiosis in cytokinesis. Okay, and remember, mitosis is also referred to as the M phase. Okay, if an organism's haploid number is 44, what's the diploid number? Haven't we seen a question like this before? What are proteins that respond to events outside the cell? Oh my goodness, is today's warm-up question on the test? I don't know, is it? Sorry about all the clicking, kids. I am reviewing my AP students' reading notes. And it's a little harder than reviewing your reading notes. They have to write 5 or 6 pages a week. And I got to make sure that they're taking notes on the right stuff, just like I do with you guys. All right, here we go. All right. Oh, sorry. I got myself confused. I gave you guys the haploid number, which means, of course, the diploid number is twice that. 88. That was a good year, 88. That was my grad year. And then external regulators, of course. No surprise there. All right, did that question. Okay, I'm going to bump this up to 2 minutes and 25 seconds for these questions. I want to make sure that we finish early enough for me to take a break before my office hours. Okay, here we go. True or false? Centrioles. Hold on a second. Let me move this out of the way. Centrioles appear, spindle fibers begin to form, and centromeres divide during interphase. The blanks help separate the chromosomes. Which phase has the chromosomes being pulled apart? 3 questions about cell division. And y'all, if you like the Beatles and especially Paul McCartney, check out his band Wings. That's what I'm listening to right now. They are awesome. It's like the Beatles, but a little bit more funky. All right, here we go. Okay, so true or false, centrioles appear? No. That stuff happens in prophase, okay? The spindle fibers help separate the chromosomes. Remember, they attach to the centromeres, and the centrioles, which are those licorice-looking things on the edge of the cell, act kind of like fishing poles. And if you remember the video, the crash course video, additionally, it's really related to some proteins that are kind of pulling from the centrioles, and they pull those chromosomes apart in anaphase. Nice. All right. All right, here we go. Cyclins are blanks that regulate the blank blank. What can occur during prophase 1 of meiosis? Really weird. Okay, guys, about 30 seconds. All right, here we go. Okay, cyclins are proteins that regulate the cell cycle. And again, remember, cyclins are internal and external regulators as well. And crossing over can happen during prophase 1 of meiosis. And of course, we already talked about why it is important. Okay, blank is a period of cell growth in between cell divisions and includes the G1, S, and G2 phases. In all forms of cancer, certain cells fail to respond to the blanks that regulate the cell cycle of most cells. An organism's gametes produced by blank have half the number of chromosomes and are different than the parent cell. All right, you guys, about 30 seconds. Remember, if you guys have any questions related to this content after I show you the answers, just raise your hand, unmute yourself, send a chat, whatever you need. Okay, this is an opportunity for you guys to get information that you might not already have. All right, here we go. Okay, those are all part of interphase. It's the signals, okay? In all forms of cancer, certain cells fail to respond to these signals that regulate the cell cycle. And of course, those signals are initiated by cyclins and regulators. And of course, meiosis produces cells that have half the chromosome number and are genetically different than the parent cell. Of course, that is assuming that crossing over took place, and you can always assume that it did. Here we go. 4 question items. Body cells are produced by blank, while sex cells are produced by blank. True or false? Excess oxygen for the cell is a problem associated with cell growth. During plant cell cytokinesis, what forms rather than a cleavage furrow? All right, about 10 seconds, guys. And again, if these questions are easy for you, take tonight off. Hit it again tomorrow night. Hit it again Friday morning. Maybe hit it a little bit at lunch. We will have some time also to review before the quiz on Friday. All right, body cells are produced by mitosis. Sex cells, reproductive cells, gametes are produced by meiosis. Excess oxygen is not a problem associated with cell growth. Lack of oxygen is a problem. because remember, it's more difficult to bring things through the membrane and also get things to the part of the cell that you need. It's kind of like a big city. It's harder to get around in because it's just bigger. And then a cell plate and then eventually a cell wall are formed instead of a cleavage furrow, and those are in plant cells. All right, during metaphase, the blanks line up in the middle of the cell. What's the correct order of the mitotic phases, or the phases of mitosis? Which phase of interphase represents preparation for mitosis? And which phase of the cell cycle do chromosomes become visible? All right, here we go. During metaphase, the chromosomes line up in the middle of the cell. Prophase, metaphase, anaphase, telophase. Of course, the only difference between mitosis and meiosis is prophase 1, metaphase 1, anaphase 1, and then the same go through the second phase. Let's see, the G2 phase is the phase of interphase that represents preparation for mitosis. So remember, G1, cell growth. S phase, double the DNA. G2, prep for mitosis or meiosis, okay? And then it goes right into prophase, okay? And then it's the M phase of the cell cycle that chromosomes become visible. So remember, during prophase, it's kind of that transition from the G2 phase into prophase. The chromatin, remember, chromatin is DNA that's wrapped around those histone proteins. It's the first level of organization. The chromatin starts to get organized, and you actually can see chromosomes. And the reason why they form chromosomes is because it's easier to equally separate the replicated DNA. If it's this big, messy ball of chromatin yarn, it's going to be really difficult to pull it apart and to make sure that each daughter cell ends up with the right amount of stuff. Okay? All right. Where are internal regulators responsible for regulating the cell found? Where are those regulators found? What's it called when cells lose their ability to control their growth rate? Chromosomes can form blanks during prophase 1 of meiosis 1. The scientific word for replicated or identical chromosomes is what? Okay, about 30 seconds. All right, here we go. Okay, so internal regulators found inside of the cell, external regulators found outside or externally from the cell. Cancer, definition of cancer is when cells lose their ability to control the growth rate. And remember, this is particularly dangerous because if you have negative mutations, those cells start to spread and they can create a tumor. And remember, sometimes tumors break off and then they spread and they infect other tissues as well. So, really, cancer cells are cells that have lost their ability to control their rate of growth. Chromosomes in prophase 1 can line up. And remember, they kind of line up like this. I'm sorry, I'm only showing this to the people that are in the meeting. They kind of line up, and this is just the top of the X's. And then this chromatid of this chromosome, and these are homologous chromosomes, crosses over and it exchanges the DNA or genes with this part of the chromatid with the DNA or genes of this part of the chromatid, creates new gene combinations, which could lead to new characteristics. And then, of course, those replicated identical chromosomes, or the pair of chromosomes is another way to look at it, are called homologous. All right, last question. So, we forgot this one up here. Unlike mitosis, meiosis creates 4 blank blank haploid daughter cells. What is a characteristic of those daughter cells? Different than mitosis. What process creates cells with one set of genes? Give you guys 2 minutes for this one. All right, 30 seconds. All right, here we go. All right, so they're genetically different, and the way that they become genetically different is through crossing over. And meiosis produces cells with one set of genes, and that's important because you're going to get the other set of genes, the other chromosome of the pair, the other DNA of the pair from the other parent. And that's why you are combinations of traits from both of your birth parents. Here we go. Last 4 questions. During mitosis and meiosis, the blanks functions by attaching to and separating the chromosomes. During the preparatory phase blank for mitosis and meiosis, the cell grows and prepares for division. Meiosis has blank cell division, or is it more than one cell division? Draw crossing over or recognize what crossing over looks like. And if you can identify what the exchange of alleles is on those chromatids that cross over, you're a rock star. All right, you guys, about 30 seconds. All right, here we go. The last answers to the last questions. All right, let's see. During mitosis and meiosis, the spindle fibers or the microtubules, remember they attach to the centromere and they pull them apart during anaphase. Let's see. During interphase, the preparatory phase, interphase for mitosis and meiosis, the cell grows and prepares for division. And the reason why you know it's interphase and not G1 versus G2 is because, remember, cell growth happens in G1, prep for mitosis or meiosis happens in G2. So if it's talking about both those things, it's talking about G1, S, and G2. And meiosis has two cell divisions. Obviously, the second one is what cuts the chromosome number in half. And then it's these letters. Let me escape real quick and do this for you guys. It's got the same thing in the book. So it's these letters. So these capital letters represent dominant alleles. Remember, alleles are forms of gene. Most genes have two forms, just a dominant and a recessive. There are a few exceptions. There are some genes that actually have 3 and 4 genes. These lowercase letters are recessive alleles. So crossing over combines recessive alleles with dominant alleles and dominant alleles with recessive alleles. And of course, if that leads to a new protein or a new characteristic, and more importantly, if it gives that organism a new characteristic, an advantage in nature, that can lead to evolution, that can lead to success in a changing environment, et cetera, et cetera. All right, let me pause my music real quick. Does anybody have any questions for me? Questions about these? Raise your hand if you're feeling confident about Friday. Don't be shy. I know you guys prepare. Nobody's going to raise their hand and feel confident. Okay, that's fine. All right, you guys. I will see you on Friday. Remember, if you want to meet during office hours, I still have openings this afternoon. Send me an email, and we can schedule some time later on this afternoon. But until then, I will see you guys on Friday.